Hi class, and welcome to the digestive system. This will be chapter 26 in your textbooks. We'll go over the general structure and functions of the digestive system, and hopefully this is a good review or supplement to all the digestive organs that you're learning in lab. So the digestive system has two categories of organs. First of all, those of the GI tract, which are listed here on the right side, and those of the accessory digestive organs. The GI tract organs are all connected with one long continuous muscular tube, and that's often called the GI tract, the gastrointestinal tract, the gut. It begins with the oral pharynx and ends with the anal canal with all the structures in between. And then the accessory digestive organs are connected to the GI tract but are not a part of it. So they will secrete substances that go into the GI tract, or they'll help mechanically break down the food. So your salivary glands, your teeth, your tongue, the liver, gallbladder, and pancreas are part of those accessory digestive organs. The digestive system functions in ingestion, which is the introduction of materials into the mouth, motility, which involves voluntary and involuntary contractions that mix and move materials through the GI tract. The two types of motility or movement are peristalsis and mixing. Peristalsis is the ripple-like wave of muscular contraction that forces material to move further along, and mixing is the name for the churning movements in the small intestine that will disperse ingested materials and then combine them with digestive secretions. These functions are also listed here with this nice diagram on the right. It tells you the different functions of the digestive tract and where the majority of these functions will occur. Secretion involves the release of mucin or other fluids like acid, bile, or digestive enzymes into the GI tract, whether that's in the mouth, the small intestine, the stomach. Digestion itself specifically means breaking down larger food items into their smaller molecular structures. Mechanical digestion physically breaks down materials. This includes mastication, which is chewing, and chemical digestion breaks down ingested material into smaller molecules using enzymes. Absorption involves the movement of molecules across the GI tract epithelium and into the blood or lymph tissue. And elimination involves defecation of indigestible waste products as feces through the anal canal and anus. So we'll start with the oral cavity. It is lined by non-keratinized stratified squamous epithelium, and that epithelium is really good at helping kind of give a protective layer and resist abrasion to all the food that enters the mouth. The oral cavity boundaries are the teeth and lips anteriorly, the oral pharynx, which is part of the throat, posteriorly, and the hard and soft palates superiorly. There's two regions. The vestibule is between the cheeks or lips and the gums, and the oral cavity proper is central to the alveolar processes of the jawbones. Mechanical and chemical digestion occur in the oral cavity. So chewing is the first part of mechanical digestion. During chewing, your salivary glands will produce saliva, which is a mixture of water, mucus, and salivary amylase, which is a specific enzyme that helps to kill bacteria and also start the process of digesting and breaking down starches into sugars. Here are the three salivary glands. Saliva is important for moistening ingested materials to become a slick bolus, we call it, of food. It moistens, cleanses, and lubricates the structures within the oral cavity. It begins the chemical digestion of carbs with amylase. There's an antibacterial action with the enzyme lysozyme, and it dissolves food so that the taste receptors on the tongue can be stimulated. And here are the three main salivary glands. The parotid lies on top of the masseter, the sublingual is underneath the tongue, and the submandibular is underneath the mandible. So here are the three different salivary glands. The submandibular gland is not the largest, but it secretes the most saliva. All of the glands in general produce about 1 to 1.5 liters per day of saliva. The pH is slightly acidic, but actually it's pretty normal. It's under parasympathetic control with cranial nerves 7 and 9. So again, the parasympathetic nervous system is your rest and digest division. So an increase in parasympathetic activity will increase saliva secretions. You have two sets of teeth that develop and erupt in the normal lifetime. Your baby or deciduous milk teeth, and then your permanent teeth replace those baby teeth. You have different types of teeth. Your incisors are the most anterior. The canines are pointed for puncturing and tearing. And then you have your premolars and the molars. And we divide your teeth into quadrants. So each quadrant will have two incisors, one canine, two premolars, and three molars. The wisdom tooth is the third molar. Many of you probably have had your wisdom teeth removed. I have not. They were really painful when they came in, but apparently there was enough room for them in each quadrant. The general histology of the GI organs, the layers of the GI tract are called tunics, and there's four of them. From deep to superficial, they're the mucosa, which is the innermost layer. Then we have the submucosa, the muscularis is the muscle layer, and then the adventitia or the serosa layer. And you can see these four layers shown here. So these are the four general layers that are the same throughout the GI tract, going from the esophagus to the anus. They will differ variably as they travel through each organ. The mucosa is the innermost epithelium. It has an underlying areolar connective tissue called the lamina propria, and then its own thin layer of smooth muscle called the muscularis mucosae. The submucosa can be areolar or dense irregular. It has lymphatic ducts, mucin secreting glands, all the blood vessels and nerves are included in the submucosa, and we call it the submucosal nerve plexus, all the nerves within the submucosa. The muscularis is composed of two layers, the inner circular layer, which will constrict the lumen and form sphincters, which open and close throughout the GI tract to allow the passage of the chyme and bolus food to move along. And then the outer longitudinal layer shortens the tube. Nerve fibers and associated ganglia between the two layers of muscles is called the myenteric nerve plexus. The adventitia or serosa is the outermost layer, and adventitia is made up of areolar connective tissue with collagen and elastic fibers. Serosa is the same, but is covered by a visceral peritoneum layer. The enteric nervous system is the digestive system's own nervous system. So your GI tract has its kind of own nervous system. It's in constant contact with your autonomic nervous system and central nervous system, the brain and spinal cord. Its two major components are the myenteric plexus, which controls the digestive tract motility or movement, and the submucosal plexus senses environment in the lumen, regulated GI blood flow, and epithelial cell function. The pharynx is another word for the throat. The oropharynx and laryngopharynx have non-keratinized stratified squamous epithelium lining. The superior, middle, and inferior pharyngeal constrictors within its walls help to swallow, and cranial nerve number 10 helps to innervate those pharyngeal muscles. The branches of the external carotid artery supplies blood to the pharynx, and the internal jugular veins will drain the pharynx. The esophagus then is the tube that connects the pharynx to the stomach. Once the teeth and your salivary glands have completed initial processing, food is swallowed. It takes about 6 to 8 seconds for the food to travel down the stomach, the esophagus, and into the stomach, and we call it a bolus at this point. The pharynx is an area at the back of the throat that connects the nose and mouth to the digestive tract, but also the respiratory tract, which is the trachea. Swallowing, there's 3 phases to swallowing: a voluntary phase, which you control; the pharyngeal phase, which was when the bolus travels into the oropharynx. The soft palate will elevate to close off the opening to the nasopharynx. And then the esophageal phase, which is where the bolus travels down the esophagus. There are 25 pairs of muscles that are helpful in swallowing. And you can read through where those muscles are located within each phase. The esophagus here we have are lined with non-stratified squamous epithelium. It has 2 layers of muscle, circular and longitudinal. Waves of these muscular contractions, called peristalsis, move food through the digestive tract. Contractions of the muscle moves the bolus to a valve called the lower esophageal sphincter, where the esophagus joins the stomach, and that valve will open to allow food to pass into the stomach and hopefully close to disallow food from regurgitating back up into the esophagus. The stomach then is a J-shaped muscular sac. It's a storage sac. It has thick expendable walls located in the upper left side of the abdominal cavity, just below the diaphragm. The stomach is involved in mechanical and chemical digestion. Mechanical digestion occurs when the stomach walls contract strongly, mixing and churning the food. These contractions are responsible for the growling noises our stomach makes when it's empty and you're hungry. And the functions of the stomach include storage of food, initiating the digestive of proteins, killing bacteria with a strong acidic content, and moving the food into the small intestine. The stomach itself, its lining has thick, wrinkled mucous membranes called rugae, composed of epithelial cells. The membrane will be dotted with these gastric pits, which are kind of deep invaginations or openings. And these pits are the open end of the gastric glands that secrete all of the fluids that make up the acidic digestive fluid with hydrochloric acid, enzymes, and mucus. Mucus neck cells secrete mucus to help lubricate the stomach and prevent damage from the acidic environment. You should know these different types of cells in the stomach, in the gastric pits. The parietal cells secrete hydrochloric acid, which helps keep the stomach acidic and helps to activate pepsin. Chief cells secrete pepsinogen, which breaks down proteins, and G cells secrete gastrin, which helps to stimulate stomach activity. Intrinsic factor is also released by parietal cells, and this allows for the absorption of a vitamin B12 in the intestine, which is very necessary to produce red blood cells. Gastric juices within the stomach, hydrochloric acid causes these juices to have a pH of less than 2, so very acidic. And this acidity allows the stomach to denature or kill off proteins, which helps to make them more digestible. It allows pepsin to become more readily available, and pepsin is most active in acidic conditions, which it needs. Hydrochloric acid will convert the pepsinogen into pepsin, and pepsin is what is needed to help break down proteins, as you can see here, into its smaller amino acids called peptides. Pepsin, as you can recall from the previous slide, is secreted by chief cells, and it works best in an acidic environment that we said, which is provided by the hydrochloric acid secreted by the parietal cells. So these cells kind of work together to break down proteins. After about 3 hours of mechanical and chemical digestion in your stomach, your food will be reduced into a small pulp called chyme. Chyme is a thick liquid made up of partially digested proteins, starches, vitamins, minerals and acids, and undigested sugars and fats. At this point, the pyloric sphincter valve between the stomach and your small intestine will open, allowing small amounts of chyme, just about 3 milliliters at a time, which will pass into the small intestine. A peptic ulcer is any erosion of the mucosa wall of the stomach or the duodenum, which is the first part of the small intestine, or due to the penetration of hydrochloric acid. It's believed not to be due to excess acid secretion, but a reduction of the stomach to reduce self-digestion. It's usually infected with a bacteria, which can survive in very acidic environments. And the other common cause is the use of NSAIDs, such as aspirin, which can damage the mucosa and inhibit mucus secretion. Blood supply to the stomach is from the celiac trunk, which delivers blood to your stomach, spleen, liver, the duodenum, and the pancreas. The small intestine then finishes chemical digestion and is responsible for most nutrient absorption. It finishes digesting carbohydrates and proteins and will begin the digestion of fats. And ingested material spends at least 12 hours in the small intestine as chemical digestion and absorption are completed. There's 3 parts of the small intestine. The duodenum is the first section, the smallest section, the jejunum is the middle section, and then the ileum is the remaining longest portion. The ileum will connect with the large intestine via the ileocecal valve. The small intestines are about 12 feet long with a very small diameter, much smaller than the large intestine, and the majority of all nutrient absorption occurs in the small intestines. The pH will vary from about 7 to 9 throughout the small intestine, which is much more basic than the very acidic stomach. The villi and microvilli of the stomach are important for increasing the surface area for absorption. The villi are finger-like folds of mucosa covered with columnar epithelial cells, and the microvilli are lined by a it's they are line the villi with a brush border, the microvilli, that helps to contain enzymes to help further digestion of starches and proteins. You have capillaries within the villus which absorb monosaccharides, which are single sugars and amino acids. The lacteal within the microvilli, it's kind of sometimes depicted as a green color, helps to absorb fats. And the epithelium at the base of the villi forms the intestinal crypts or crypts of Luberkuhn, and these help to secrete mucus and allow for replacement of columnar cells. They also help to protect against pathogenic bacteria. Here's a look at the submucosa of the duodenum. It has Brunner glands in it, which are found in the duodenum that secrete an alkaline or basic mucus to help prevent damage from the acidic kind that's coming from the stomach. There are some anatomical changes as we progress through the small intestine. Goblet cells become more numerous towards the ileum, and the villi become larger and more numerous in the jejunum, and also have more circular folds because most absorption occurs in the jejunum. So, absorption, once we have chemically digested the food, we must be able to absorb them into the capillary. Carbohydrates are broken down into monosaccharides, which are single sugars. Proteins are broken down into amino acids, and triglycerides into short and long fatty acid chains. Both active and passive transport are used to absorb these nutrients, which you'll learn a little bit more about in physiology. But basically, the absorption of the small intestine it breaks it down into its molecular components, and then it absorbs it through the epithelial cell of the villus, and then through diffusion into the blood capillaries. The missile stands for fat, and that is going into the lacteals. The blood supply to the stomach is due to the superior mesenteric artery, which delivers blood to the duodenum, jejunum, ileum, as well as the pancreas, cecum, the ascending and transverse colon. Where do the absorbed nutrients go? So once all the nutrients get absorbed from the small intestine, they're absorbed into blood capillaries of the intestine and they're delivered to the liver. This is known as the hepatic portal system, and this blood is very nutrient rich because they are veins going from the digestive tract to the liver and eventually back to the heart. So they're also oxygen poor. The hepatic portal system, so your liver takes all these nutrients absorbed from digestion and kind of filters them out, detoxifies them, stores a lot of the glucose so that it can be later used for body needs. The hepatic portal veins will drain blood from the intestinal capillaries, pancreas, gallbladder, and spleen. The hepatic portal system accounts for 75 to 80% of blood flow to the liver. The hepatic portal system, the liver also gets blood supply from the hepatic artery, which accounts for the remaining 25% of blood flow. And the hepatic portal artery will bring oxygenated blood to the liver so that its cells can carry out its functions. The basic scheme of the hepatic portal system, the liver histology is made of connective tissue, which partitions the liver into thousands of these kind of polyhedral hepatic lobules, which contain hepatocytes or liver cells. At the periphery of the lobule are several portal triads, which include the hepatic portal vein, the hepatic artery, and the bile duct. And the central vein is in the middle of the lobule that acts as the major drain. The hepatic portal vein will carry this nutrient-rich blood from your GI tract to the liver. The hepatic artery provides oxygen-rich blood to the liver, and the hepatic veins empty into the inferior vena cava, which eventually brings the blood back to the heart. The liver is made up of these thousands of hepatic lobules, which are functional units of the liver, and the lobules are made up of hepatocytes, which are liver cells. Hepatic lobules contain these radiating hepatic sinusoids, which are leaky capillaries lined with the reticuloendothelial cells, which are phagocytes with an immune function to digest anything harmful. Hepatocytes will absorb nutrients and produce bile. Between cords of hepatocytes are what we call bile canaliculi in green that bring bile to the bile duct in the portal triad to eventually bring the bile out of the liver. So this is what each lobule contains: the central vein, the hepatic portal vein, the hepatic artery, and then the sinusoids, which allow the blood from the hepatic portal vein and the hepatic artery to mix. The hepatocytes will also absorb some of the nutrients that are passing by in the sinusoids and store them in the liver, and the remaining blood will travel to the central vein, to the hepatic vein, and eventually to the inferior vena cava, back to the heart. Other functions of the liver are to produce bile, detoxify drugs and poisons, clean up and recycle red blood cells, store excess nutrients, and produce other plasma proteins. Liver processes carbohydrates by storing excess glucose as glycogen. It stores fatty acids used to make ATP, and it also takes care of proteins when amino acids are converted to urea, and it helps to make ATP or convert them to glucose. The liver and gallbladder are connected. They're responsible for secreting a yellow-brown liquid called bile. Bile is stored in a small sac called the gallbladder, and the entrance of food into the small intestine stimulates the release of bile by CCK, cholecystokinin, into the small intestines through the bile duct. Bile will be stored by is produced by hepatocytes in the liver and stored in the gallbladder until needed. So hepatocytes just create the bile, and the gallbladder is what will concentrate and store the bile. So people can live without their gallbladder if they get gallstones. Some will get their gallbladders removed because their liver continues to secrete the bile. The biliary apparatus is the network of thin ducts that transport the bile from the liver and the gallbladder to the duodenum. The left and right lobes of the liver drain into the left and right hepatic ducts, and the left and right hepatic ducts merge to form the common hepatic duct. So this kind of method of ducts is important. Probably you'll learn this in the lab portion too, but these right and left hepatic ducts go into the different lobes, and the common hepatic duct exits out of the liver. The cystic duct exits out of the gallbladder, and the common bile duct is the combination of the two that will take the bile into the duodenum. The pancreatic duct comes from the pancreas. The pancreatic duct and the common bile duct enter together at the major duodenal papilla to bring bile and pancreatic enzymes into the duodenum. Bile is important for digesting fats. Bile salts will emulsify fats. It will take larger globules of fat and make them smaller for easier digestion. One of the main functions of bile is to dissolve cholesterol. A buildup of cholesterol or bile salts can lead to calcification of these substances, known as gallstones. This is a look at gallstones with filling up a gallbladder. Incredibly painful because they can block the cystic duct from the gallbladder. They can be seen from an X-ray, and this is somebody's gallbladder filled with gallstones. Not good, very painful. The pancreas's role. The pancreas organ is located behind the stomach, secretes pancreatic fluid, which enters the small intestine, the duodenum, through the pancreatic duct, which joins with the common bile duct just before it enters the intestine. And pancreatic fluid is important because it contains enzymes that help to digest proteins, fats, and carbohydrates. The pancreas, if you remember, has both an endocrine and an exocrine function. 99% of the pancreas is made of acinar cells, which release digestive enzymes, and 1% of pancreas is made up of pancreatic islet cells, which release hormones, insulin and glucagon. Insulin lowers blood glucose levels, and glucagon raises blood glucose levels. So that's the endocrine function, the hormone function, and the exocrine function is using the acinar cells to release the pancreatic juices for digestion. The pancreatic juice is made up of bicarbonate, amylase, trypsin, lipase, and they're all regulated by the release of CCK from the duodenum. Bicarbonate causes the area of the duodenum to become more alkaline, which allows for better absorption and protection from acidic chyme that came from the stomach. Amylase digests starch, trypsin digests protein, and lipase digests triglycerides. The gross anatomy of the large intestine is made up of the cecum, the first part, which is kind of the sac. The appendix sticks out from the bottom of that, really no known function. Then we have the ascending, transverse, descending. Sigmoid is the S-shaped part, rectum and anal canal. We have flexures where the large intestine will curve. The large intestine is covered by simple columnar epithelium. It's larger in diameter than the small intestine. That's what gives it its name. It contains many mucus secreting goblet cells. There are no villi. It contains pouches called haustra, and the teniae coli is kind of a string of smooth muscle that helps to propel the chyme and feces through the large intestine. Also known as the colon, the main functions of the large intestine are to absorb water and electrolytes, absorb several B vitamins and vitamin K, which is important for blood clotting and the transport of calcium. Most absorption occurs in the ascending and transverse colon. Water absorption of the large intestine, sodium will be transported out of the lumen and into the blood. Potassium is transported from the blood into the intestine for removal, and this leaves an increase in solutes in the blood, which causes water to passively move into the blood from the large intestine. The large intestine also contains a large amount of healthy bacteria, which we call microflora. And these help synthesize the B vitamins and vitamin K. They also help to ferment some indigestible material used for energy by epithelial cells. It also helps to promote water absorption and electrolytes and helps to maintain healthy epithelium and protection. Defecation is just the process of eliminating excess waste. Once all the water and electrolytes have been removed, the waste material passes to the rectum, and this increases rectal pressure and starts the activity of the anal sphincters. The internal anal sphincter is involuntary, and once it relaxes, it starts the urge to defecate or pass that. The external anal sphincter is what we control, and that's under voluntary control. I'm trying to get my 3-year-old to voluntary control her external anal sphincter so she can go in the potty and not her pull-up. The autonomic nervous system, as you remember, helps, whether it's sympathetic or parasympathetic, to control the digestive tract. Sympathetic control inhibits GI secretion, inhibits motor activity, and contracts GI sphincters so that food does not flow through the GI tract. Parasympathetic control, which is controlled by vagus nerve, cranial nerve number 10, will stimulate GI secretion, stimulate motility, and increase blood flow to the GI tract. Neural control, you have 3 phases to gastric secretions and motility: cephalic phase, the gastric phase, and the intestinal phase. The cephalic phase is initiated by sight, smell, taste of food, controlled by the vagus nerve, and will begin secretion of pepsinogen and hydrochloric acid in the stomach. The gastric phase is when the stomach is stretched, which will stimulate vagus nerve and stimulates gastrin and pepsinogen secretion. And the intestinal phase inhibits gastric emptying and acid secretion. It promotes duodenal activity, giving the intestine time to absorb the food. And that is the end for this chapter. Thanks for listening, guys. And as always, let me know if you have any questions.